Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about the principles associated with mapping complex traits and how we do this in both crosses and using genome-wide population studies. In this video, we'll elaborate on the previous one to look at actually what's done in genetic crosses to pinpoint the location of genes that affect or that have allelic variation affecting some sort of trait. Now again, as I said at the very end of the last video, the ultimate prediction is that if we look at many linked markers, we should be able to pinpoint the location of a QTL by where the association is strongest. And we talked about exactly what that meant in that context. So what we'll do is we'll follow the trajectory of association strength to infer the location of the QTL. Now let me illustrate this with an example so you know what I mean, because trajectory of association strength may be a little bit difficult to understand. So let's use an example from the mapping cross. We'll stick with the same example of corn that we used last time. This was a cross where we crossed a tall strain to a short strain. The tall strain was 5 feet tall. Short strain was 1 foot tall. We got F1s that were intermediate. They were 3 feet tall. And then we crossed the F1s to each other and got a lot of variation where some individuals were 5 foot tall, some individuals were 1, and a lot of them were kind of in the middle. We got that sort of bell curve. Now what we're not trying to do is we're not trying to get recombination fractions this time. This is different from what we did in the context of simple genetic traits. It's much more complicated when something is complex, so we're not going to do that. What we're just going to be doing is looking at the association of genotype and phenotype, okay, just looking at that association, and we'll look and see how strong it is. Now remember, strength is defined by the average difference between the genotypes, okay? So what we'll do is we'll genotype 5 markers rather than 2 this time, and look at how they associate with height in these corn, okay? So let's dive in. So let's say you're looking at 5 close markers to the trait. These markers are about 2 centimorgans apart from each other. So the markers recombine from each other, the alleles of these markers recombine from each other at an average rate of about 2%. Now let's say we're going to put on our divine glasses where we now can see what the actual answer will be. So our divine glasses, we see that the height gene is next to C. Now we don't know that, as scientists we don't typically know that, but for this example, let's say that we do. Would we see an association of height with the E genotype? Okay, so we know the height gene is next to C. Would we see an association with the E genotype? Well, the E gene is only about 4 centimorgans away. So sure, of course we'd see an association with it. It's pretty darn close to that gene which is contributing to variation in height. Well, we might see something like this, where the E gene, if you're big E, you're 3.5 feet tall. In this case, again, I'm using the case where all the capital letters are derived from the tall parent and the lowercase letters derived from the short parent. The big E ones are 3.5, the little e ones are 2.5. Okay, so again, we do predict some association between E and height because it's only about 4 centimorgans away. Well, what about D? Do we think there'll be any association between D and height? Well, obviously the answer is yes. Sure, but the important thing here is we see the genotypes at D are more strongly associated with height. So you see the difference here between big E, and little e was about 1 foot. Here, between big D, and little d, it's 1.4 feet. So more of the variation is explained by genotypes at D than by genotypes at E. And that's because the D marker is closer to the gene that's causing the height difference. So let's fill in the rest of these. There we go. So we see the same sort of thing here. What we see is as you get further away from the height gene, less variation is explained. So when you're 4 centimorgans out, you're explaining about 1 foot of difference in height. When you're a little bit closer, you're explaining 1.4, and when you're pretty much right on it, you're explaining almost 2 feet of the height difference. Okay, so the prediction is that we see a stronger association when we're looking at markers closest to the gene affecting the trait, which in this case is corn height. Now this has been done quite a bit, and this is often depicted using what's referred to as a LOD plot. So what they do is they use math, which we're not going to cover in this lecture, to translate this observed and expected associations to identify a likely position of the genes affecting the trait of interest. So in this case, this is looking at various traits associated with tomato, and pH here is very well explained. And you see, looking across chromosome 10, there's an area here where there's a lot of the variation in pH explained, and there's areas where it's not. Now, typically speaking with LOD plots, a high number means a more likely location for genes causing the effect. Often the cutoff of about 3 is used, that if you see a LOD score above about a 3, that means you see a statistically significant association. So what this would actually mean is, they looked at markers at all these different places, right? The markers aren't actually drawn on here. And this one right here had a very strong difference between individuals that were big P, and little p, in terms of the pH. This one had a weaker one, this one had a weaker one, etc. So we infer that a gene affecting pH is probably right around there. Okay, now it's an inference, it's not an exact pinpointing, but it's an inference of a likely location for it. So how is this different from when we were mapping simple traits? Well, the big difference now is that we're looking at markers in multiple regions of the genome associated with the trait, and a lot of them are explaining it. So this picture here is from a map of QTLs that are associated with Drosophila mating success. As you can see, there's a lot of these little peaks. This is the same sort of log plot as I just showed you. So here, this is the X chromosome, second chromosome, third chromosome. There's 3 different places on the third chromosome, at least, maybe a fourth, that are associated with variation in mating success. These little triangles that are a little hard to see in the very bottom, those are actually the locations of the markers. So what we can infer is the association of this marker right here is bigger with mating success than the association of these markers here, which is effectively negligible. Okay, same sort of thing here. Big association with this marker, negligible with that one, relatively speaking. So you're inferring from this trajectory where the likely positions are of these genes affecting the trait. Now let me give you one to try. Infer the location of the QTL by the trajectories of association here. This is basically the same principle as before, but what would you predict in this case? Here we have an association with A, B, C, and D. Hopefully that wasn't too difficult for you. What you saw is that A and D had weaker associations than B and C. B and C had similar associations. So probably, as you're looking at this trajectory of association strength, we see it getting stronger this way, we see it getting stronger this way. So you'd probably infer that the gene causing this height difference is probably somewhere between B and C. That's the difference from what we did before, right? In this case, it wasn't right at C, but it was actually between 2 of them. So you might make a log plot that looked something along the lines of this. Now, I mentioned to you before, these things are hypotheses, and there are dangers. You can actually be misled. Now let's imagine that we looked at another gene right in the middle, another marker. Now, let's say it's the marker T. What we expect is T should have a stronger association with this trait than B or C. But what if we do that and we see this? Well, what does that imply? Well, in this case, adding the T marker in there, we see that T has a weaker association than B or C. So rather than having one single large mapped effect, it looks like maybe we're potentially mapping 2 different genes that have allelic variation contributing to this. So maybe there's one near B and one near C rather than a single one near the middle. This happens quite a bit in the course of genetic mapping, something which looks like one strong association, therefore a single gene with a large effect. So you map a little bit more finely and ends up being, actually there's a couple of different genes in that same area, they just happen to be a little bit close to each other. So we don't know what makes the QTL effect. We see these effects in mapping, and these are inferences of the locations of a factor or factors that are contributing to this variation. There may be multiple genes that are close together, or there may be a single gene. But the reason I stress this, and this is very important, the QTL mapping, whether it's by cross or pedigree or by population association study, is not conclusive. It is a hypothesis of where one or more genes are affecting the trait. And ultimately, you have to go in and do other techniques to confirm that is truly where there is a gene that has allelic variation contributing to this trait. Hope that was helpful. Thank you for joining us.